quite a bit to cover after that. So, but the housekeeping stuff is here. I just loaded it. And I had to send it to you, one of these by email. I'll send the other one by email as well. Some of the files that we're going to start to see from the other textbook I'm using, the Marie, I use it because of animations. And there's some really good ones coming forward, particularly in this. We start with this chapter, as well as muscles and nerves. Apparently, they are too large to upload on Blackboard. I tried to load it in the other class, so I sent it to you by email. I mean, it's up to you. You'll see them if you get the recordings and listen to them. Just if you want to download it, that's fine. A student asked me about printing it. If you want to print it, that's fine. Sometimes there's some confusion about printing, and I address it now, about printing PowerPoints and stuff like that vis-a-vis copyright. When you're using them for educational purposes, that's not a problem. If you're not intending to sell them, or if you're going to use them to write your own book, that's a different story. But I mean, for educational purposes, we copy and photocopy things out of textbooks all the time, just because it's educated. We've already paid the fee, so to speak, by buying or furnishing the textbook for education. It's my understanding. I'm not a lawyer, right? The other thing, so I did load the Welsh Hull chapter PowerPoint, which actually for this chapter is pretty good. They don't cover something that I hope we may get to today called resting membrane potential, which is sort of the foundation about how nerves and muscles work. Whether we get to today or not, I don't know. We have whatever to cover. Additionally, I have a mini outline here that you'll see so this is on the Blackboard loaded to you. The supplemental outline, just opening it now, you'll see these kind of, it talks about the plasma membrane and different channels and blah, 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 blah. So maybe it's some stuff you had before, but useful to read it. So I've tried to effectively outline, just as I've done before, a similar chapter. It may make it, and that's the kind of stuff, sometimes it makes it easy if you got nothing better to read. That certainly puts you to sleep. So I've gotten older. I just got a new book to read. I read fiction. I can't keep my eyes open. I read on my iPad. It's awful. Nevertheless, so that covers what you'll see there. And I did send you, and we'll cover it today. I'll show you exactly where the test material ends. And I did it this way for a reason. And our test will be in 2 weeks. The reason is I want to get what the proteins and the membranes do, because it's really to understand the next chapter, and they sort of reinforce each other. Once you understand how the membranes work and how the cells are joined together, it'll amplify and sort of reinforce how things move across membranes, which is really the next major topic. And so while I'm talking about that, in the meantime, it'll sort of be reinforcing what the role that the proteins have as well in the membranes. So that's basically the sum and substance of what we're going into. So I figured I'd give you that little introduction for a couple of minutes. And I know roughly where we left off. And I know we covered the proteins. I did want to amplify some points, but I don't recall if we got to enzymes. I don't think so. I always look at the last recording at the very end to see where we left off. So we can start. So here we are. So this is the enzyme and enzyme activity. It really, you'll see that Welsh Hull PowerPoint 3 covers this, and PowerPoint 4, which I'll load for you, covers these things pretty well. These are very physiological. Everything is governed by enzymes. They're globular proteins, so the shape is so important. And we'll see all the ones and you'll see all of them in the membrane, the roles they do, and there are even these in the membrane. But every biological reaction, almost without fail, requires some kind of enzyme. And enzymes have a very specific definition of what they do. It's called they lower the energy of activation. In short, and it's something you'll see right after this when we talk about DNA and RNA and ATP. Every time we want to do something in the body, we have to put energy into it. Anytime we have two ingredients that we're either going to make them come together, or one ingredient we're going to pull apart, or 6 ingredients to put together, we have something called the energy of activation. Okay? And so to determine whether or not the overall equation stores energy and makes bonds, or releases energy and breaks bonds, we sort of do the math at the end. But you need to put a certain X amount of energy in, 4 ATPs, 10 ATPs, 20 ATPs, because that's the currency that we use. So the whole idea is about what enzymes do is to make it happen faster, they lower the activation energy. They make it easier for this to occur. Very, very specific. But enzymes are great because they don't get used up. They have a very long lifespan. And like any protein over time, they're probably going to denature. But as you can see here, millions of reactions per minute, and we use them over and over again. And so it's just, when you're talking molecularly in the human body, with all those, with billions upon billions of equations and reactions going on, this makes it very rare. A little drop of enzyme in something. In the old days, when they used to drink lactate milk. Maybe they still have it. You put a couple little drops of the enzyme into regular milk, shake, and all of a sudden you could feed it to somebody who was, like one of my kids who was lactose intolerant at the time. It was like, it worked almost magically. That's an enzyme, very, very fast. So enzymes in general, okay, I don't use the terms, you have to know holoenzyme and apoenzyme. Again, you'll have different people who may ask you that question. For some of you, when you have board or licensure exams, terms like this get thrown out. That's why I just present them to you. So it'll ring. Holo means whole enzyme. Apo, protein. So it's the P for protein. To me, they're dumb. Okay, an enzyme is an enzyme. It's made of a protein. Whether it's the whole thing or just the protein portion. What they're getting at, these enzymes are very shape-oriented. So effectively, they look kind of like this. If you get something like that, and piece that fits in the enzyme has a couple of fancy names, and they're interchangeable. You can see the term either ligand or substrate. It'd be nice if they said the thing that fits in the enzyme. This part of the enzyme where it fits is called the active site. That's where the activity occurs. So it's active. To make that active site, sometimes, and maybe the best way to represent it is using a different color, it sometimes will require a piece of organic material to sort of make that site complete. We often call that a coenzyme. You and I have a different name for it. We call them the vitamins. So coenzymes by definition are organic, and typically we call them vitamins. Even if it doesn't sound like it's vitamin B3 or vitamin B6 or whatever, it's like folic acid, it's really in that category. They basically work to create an organic portion of the enzyme, and a lot of times they require a mineral, let's say zinc or iron. We call those cofactors. And you and I, and they're inorganic, you and I call them minerals. So and they are by definition, something we touched on a little bit before, they are essential, meaning you have to acquire them through diet. We can't make the enzymes, we can't manufacture the minerals, and you don't need a whole lot of them. I mean, this is this is Kevin. I should take one a day, and I don't. I used to be a little bit better about taking these things. My wife and I are getting lazy. So we used to get a big bowl of supplements and enzymes and stuff like that. But now I try to remember to do it. But really, it's a good idea. And they're so easy, and then a lot of times they're designed for men or for women at different ages. So these are they don't really hurt you at all. Typically, the enzymes, the vitamins and minerals you get like that, it'd be very hard to overdose on something along those lines. that are there. I'm not saying to gobble down a bottle. We utilize the suffix ASE. So lactose intolerant, the enzyme is lactase, that little thing in the dropper bottle that we used to use 45 years ago, whatever, however long it was. That kind of a thing. And so it tells you what it does. Hydrolases work with water, hydroxidase. It could say oxygen when it really has to do with like redox reactions in chemistry. Oxidation when you lose electrons. I don't want to get into that for you guys. It's really, that's a different, I mean, if we were doing, with my advanced anatomy and physiology students, that's the kind of stuff we were talking about. Because it's, it makes something that you really don't have to know, at least to that extent, more complex than you need to. Here's the lower activation. They talk about priming the reaction. I'll buy that. And what it basically, well, the things that involve is that substrate, that thing, binds. We call it now, we have a fancy term that is different. They usually talk about the lock and key bond. The problem with lock and key, the implication was once you put it in there, like sometimes the key will get stuck in the lock, you can't get it out. Now we use something we call induced fit. It really means is that ligand or substrate gets closer to the active site, they both start to change. And that's what facilitates the things either coming together or going apart. It's all about the shape. When something gets denatured, we talked about pH, and we talked about temperature, denaturing proteins. Effectively what happens, it will cause that active site. Now you got that thing sticking out here again, but all of a sudden the active site now starts to look like this. Will fit. I'm doing it two-dimensionally, but effectively that is what happens. These proteins effectively change dramatically. Again, whether you're cooking the egg or got a steak on the grill, any way you want to think about that, that's where the alteration occurs. And they always show this classically. So here's without the enzyme, you have to put in a substantial amount of ATP just to get it going. Where with the enzyme, comparatively little. Every time I've heard this or done this lecture, it's like going skiing and you could walk yourself up to the top of the mountain. You'd be putting in a lot of energy to get there. But here, so the enzyme is the ski lift. If you want to think, it makes it the ability to accomplish the task that much easier, that much more likely. Whatever idea works for you, just remember lowers energy of activation. Boom. And they're just showing you some examples of active sites. I don't get into it. It was pretty well done, and I think it's either your textbook's chapter 3 or chapter 4. Enzymes play a really interesting role in negative feedback, and it has to work with changing that active site, but not damaging the enzyme, so to speak. And I'll see if we get in, and we may get into that later. I don't want to put it now. It's a little bit more of a sort of an advanced approach, and I want to get the basics more out of the way because it alters this. Here's the active site. So amino acids, 2 of them here, put them in the appropriate proximity, and it facilitates the formation of the peptide bond forming, and water is released. And this is the last part of the biochemistry. This is the part I was doing with all the classes today. And finishing up, so it kind of puts us into the right frame of where we want to be, more or less, maybe a little ahead. Nucleic acids. When we did the proteins, they were amino acids. They had nitrogen. The other major nitrogen-containing component are called nucleotides. They have what's called a nitrogenous base. All that means is it's a series of carbons with nitrogen incorporated. You learned about that. When you were in lab, you talked about ground substance, you talked about connective tissue, you talked about matrix. That ground substance, whether it's in connective tissue, whether it's in cartilage, even whether it's in bone, is basically a carbohydrate with nitrogen. You stick nitrogen with a carbohydrate, it strengthens it. So we use nitrogenated carbohydrates. The amino acids also contain nitrogen. Nitrogen is a pain in the neck. I mean, we utilize proteins constantly. We break them down, we build them up. We do the same thing with DNA and RNA and these nucleotides. The problem is, everybody, we have a limited ability to tolerate accumulation of nitrogen in our system. So all that nitrogen has to be excreted with a limited amount that can be in your bloodstream. That's what your major job of your kidneys, I think. I mean, you can talk about pH, you can talk about water balance and other electrolytes. If they don't get rid of the appropriate amount of nitrogen, you are in big trouble. That's when your kidneys start to fail. I have been there. It's not a pleasant thing. About 20 years ago, I got very sick and ended up in ICU. And what brought me there was my kidneys decided, oh, we've had enough. We're going to shut down for a while. It came from an infection. It wasn't that. The tests they use to evaluate that are nitrogen. They look for something called blood urea nitrogen, or BUN, and they look for something called creatinine, which we're going to cover in muscle. Those are nitrogen waste products that you only can have a little bit of bloodstream. Just to give you an idea of the importance of the nitrogen. So, nucleic acids, very, very specific in that regard. They are the polymer, where proteins or polypeptides are the polymers, and amino acids were the monomers. There just happen to be 20 R groups. Here, you have 4 monomers, okay, for DNA, 4 monomers for RNA, with a slight difference between the monomers that I'll get to in a second. The polymer is DNA and RNA. So we call them, the monomers we call nucleotides. And by definition, you're going to see they have that nitrogenous base. Now we get 5 carbon sugars and a phosphate. That's the buildup that's there. And depending on what that sugar is, if it's deoxyribose, we're going to make DNA. If it's ribose, we're going to make RNA. And it's amazing, that tiny change structurally, by not having an oxygen in 2 nearly identical molecules, is the difference between RNA forming a linear molecule that has the ability to leave the nucleus and communicate all over the cell, versus DNA, the single most important and largest of all the macromolecules that we have, being able to allow cells to replicate and how cells to express themselves in making proteins. It's miraculous. It's magical. So, DNA. It not only is it the genetic blueprint for making all our proteins, and really there's not a big difference between any of us. Okay, I've read a couple of articles recently about that as I was preparing to do this. I mean, over the years, it varied between 97 to 99% of our DNA, depending on the author you read, is the same. The vast majority of it doesn't make proteins and doesn't make genes that effectively make us who we are. Maybe at 1%, 1.5% on average is why you look one way, I look one way, why you're a good athlete. I've got a vertical leap of about 1/2 inch. You know, I'm never going to be Scotty Scheffler or Tiger Woods with a golf club. Okay, unless I'm carrying their golf club and walking with them. That's just the way it works. Okay, there's not a whole lot of difference between people. It would be useful in the society we have today, I'm editorializing, if we all learn to get along just a little bit better. In some ways we do that better now, and in some ways we do that worse, but that's not a discussion for a science classroom. So on with the show. So what does this all mean, all this gobbledygook stuff about bases and riboses and stuff like that? So if we're talking about DNA, we have the base. Let's just put the base, the sugar, the phosphate. The bases have different categories, okay, as you can see here. Okay, so the sugar has to be deoxyribose. The phosphate is generic. You're going to see when we write it, it's got a formula, which you don't have to really remember. PO4 with a minus charge on it. And the base can be different. There's 4 different bases. And you need to know the categories. Purines are adenine and guanine, and they're very similar, and we use them a lot in the body. That's why they're really important, because it's not all just DNA or RNA. And I won't draw they basically consist of 2 of these carbon-nitrogen rings. They're better drawn than I have. In DNA and in RNA, they're the same. The pyrimidines, which don't have 1 ring, they have 2 rings. They have 1 ring rather, not 2. And in DNA, they have specific names: cytosine and thymine. And you'll see when we get to RNA, and we always write this, the A's, you probably had all this, the G's, the C's, and the T's. And the only difference is when we get to RNA, thymine is replaced by something that looks almost exactly like it chemically, called uracil. At the same time, we change the sugar from deoxyribose to ribose. There will be on the exam. Here's the answer key. Here's the answer key. DNA only, RNA only, both DNA, RNA, neither. Got it? Adenine, it's in both. Uracil, it's in RNA only. Ribose, it's in RNA only. Deoxyribose, in DNA only. Got the idea? That's kind of like a modified match. I'm giving you no extra charge. Donations are great. We got to put a tip here. I can't be any more transparent, using the modern expression for not BSing you, than that. So, voila. What does this all mean? Okay. The whole idea, the magic of DNA, this unbelievable molecule that we know now is like a biological zipper. It opens and closes. It opens to either make more cells to replicate, or it opens and uses one of the strands, and we call it a template, the model, to basically express itself in making proteins and something, and make RNA to express itself in making proteins. It's magic. Everybody knew. They knew how important it was. Roughly at the end of World War I, scientists were very, very well aware of this. And they knew that the A's always were in they had the same number of A's as you had number of T's, and the same number of C's as you had of G's. So A's and T's were equal, C's and G's were equal. They couldn't interpret it because they didn't really know the structure. That's why it was so darn important. So it wasn't that it wasn't like 50% A and T and 50% C and G. Those varied, but when you looked at them individually, it was proportional. So they were equal amounts. We know now that because of the way it's cross-linked, you'll see, and we'll have seen it, or we'll see it again. What happened was, unfortunately, at the end of World War II, everybody was trying to solve that. At the end of World War I, rather, they were trying to solve the problem. And unfortunately, World War II occurred. When war was upon Europe and subsequently the U.S., all the scientists and doctors who were studying this basically would get involved with the war, for whatever country or nationality they are, and whatever side of it. Exactly. So at the end of World War II, all of a sudden everything changed, and then some scientists went, that's what they started to develop rocket programs, space exploration, blah, 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 blah. What a lot of them do is they started looking to figure out what DNA was. They weren't really scientists. And they had a group in England with an American, and I forget which one is which, called Watson and Crick were their famous names. They had another doctor with them whose name I don't remember. And they had a lady doctor with her named Rosalind Franklin, who unfortunately never got credit for really solving the puzzle. She came up with a vital piece. In our country, out in Caltech, I had a guy named Linus Pauling, who was a two-time Nobel Prize winner, was also studying. Just so happened Watson and Crick got it first. They got it because of Rosalind Franklin. If you did not know, molecules like DNA come in the form as a crystal. Okay? She did what was called X-ray, basically crystallography or X-ray diffraction. She would X-ray crystals and try to analyze their shape. The trouble is with this twisted ladder, the ability to open and close, which they weren't aware of, she couldn't get it. It was blurry. You take an X-ray. I took X-rays in my office. If the person's moving, it's blurry. That's why they hold your breath. Thank you. They tell you to breathe. You have to have a test X-ray or anything like that. You don't want you to move. It's the reason. She figured, I know what I can do. I can freeze it because it'll stop moving around. And it wasn't like putting it in the freezer. She put it in like liquid nitrogen and took the X-rays. She discovered that there was enough room Here's the complementary base pairing, A's and T's, G's and C's. She discovered that there was enough room in the middle here, because she saw the structure of the double helix, actually a spiral staircase, and the fact that all the steps were the same width. And she knew by analyzing that number repetitively that it meant that you would always have a 2-ring with a 1-ring, 2-ring with a 1-ring. And so what she did not know, because that wasn't her end of it, putting it together, but she basically communicated that effectively. They said she did that like overnight in a couple of days, and she came down wherever they were gathered, either in a meeting or just in a common area or a lunchroom, and basically told Watson and Crick, I just did this freezing, came up that this was the actual dimension, and interestingly, it looks like you could fit a 2 and a 1 together. And Watson and Crick went upstairs and took Tinker Toy models. Just Google it, you'll see the original model. They're right next to it, famous picture. Made it. And 5 years later, they get the Nobel Prize. They never gave her any credit. She got her credit posthumously. She never got the Nobel Prize, but at least she was recognized as having played a really important role, which she did. And unfortunately, it underscored something that was extraordinarily prevalent then. Thankfully, a little less, but still prevalent now, that women in medicine and science do not get the credit they should. It's tragic, horrible. I can't think of enough bad words. I want to fear that, but that's the nature of the beast. So that's the very famous story, and you can see they came up with this model. And effectively, what they found, and those little dots are the key. They're called hydrogen bonds. We learned about that with water. It's a weak magnetic attraction, so it can open and close. Hence the biological zipper. The fusion of the phosphates and the sugars make a pivotal difference, okay? Because they create what we call the backbone. Each one of these is fused to the other, and then you'll see the sugar of the negative. The sugar here fuses with the next one here and the next one here, and we call that it's called the sugar phosphate backbone of both of these. And we see it both in RNA and DNA, and it really plays a pivotal role in organizing the molecule. Look at this picture. Without having to go through it, you're going to see in a subsequent animation that I'm not going to play, but I'll go through. Look at the positioning here of the ribose. Look at the molecule here and the molecule here. This one, if you will, is right side up. This one is upside down. And that is a pattern that goes throughout. And what you will learn about it, what we learned about with DNA, is that, and why it doesn't matter what strand we use to make proteins. And when we replicate it, we take apart the two strands and we make new strands to each one of those. That's the way it's been. We've understood that for a long time. Effectively, there's directionality. One strand is right side up and one side is upside down, even though we're identical. So it doesn't matter which strand you use as a template, as a model, you're going to make the same number of proteins, the same ones. We have a fancy name for this that you may have heard. It's called anti-parallel. So parallel just means they're going in the same direction, except one's going up and one's going down. Parallel in this is it's not like geometry parallel. You looked at, in lab, you looked at slides of tendon. Tendon and ligament have dense regular. What meant by the regular fibers, those collagen fibers, they were parallel. They were wavy, but they were still in parallel, means they were wavy, but basically identically, directionally speaking. Same thing here. It's not like geometry, two parallel lines and a transversal. That whole thing with all the angles. I took that class. I was a math major first. Just everywhere. So voila. That's the idea of being anti-parallel. And the fact that we take one strand of each when we make new cells, it kind of keeps the strands very fresh. We call it semi-conservative replication. I don't know if we cover that here, but I talk about it a bit in micro. Semi-conservative means you're half. You're conserving half of the original DNA and making half new. And what that is, it keeps the DNA relatively fresh. And it's a little different. Cloning, you don't do that. And what happens when you clone organisms or you clone cells, they begin to fade. It's like a photocopy of a photocopy of a photocopy. Where here you always have a certain freshness. So it's a subtle difference between that and the other. Voila. So RNA basically is the way the rest of the cell. Here's DNA is huge. It can't leave the cell. The nucleus of the cell, I beg you can't leave the nucleus. The nucleus is a large structure, and as you will see, it has something called nuclear pores. They are designed to let nutrients flow in, waste products flow out. DNA is contained within there. There's only two areas in the cell where we find DNA. We find it in the nucleus, and as you will learn, but not likely today, they also find it in mitochondria, and it's a different constructed kind of DNA. And additionally, what that means is the DNA has to communicate by allowing the RNA to take this material out of the cell. And it does so. There's 3 different RNAs that are done for this. And this is a process. DNA, when replicating, is pretty straightforward. But when we're making proteins, it's a very complicated process whose names you may have heard. DNA making RNA is called transcription. It makes messenger RNA. It is transcribing its message, its actual batting order for the amino acids and the instructions on how to assemble. It's like that thing when you get the new package and how to assemble and all that. That's transcription. It's transcribing that message into the language of RNA, which means we have the U rather than the T, even though they're very close friends. So transcription isn't sort of generic instructions. It's very specific. That's what a transcription is. In the days, I do not know what they do in hospitals now. I haven't operated in 25 years. 25 years, whenever you finished an operation in a hospital, you dictated a report. So whether it was a phone or a dictating station or an old tape recorder, you dictated what is called the operative report. This is what I did during the operation. These are my findings, blah, blah, blah. It took 5 minutes. And we did them almost automatically, mechanically. And then they would go to a medical transcriptionist, someone who would receive that recording either immediately or on a disc, and they would type it up. And if they didn't know the spelling, they would spell it phonetically, and if it was a complicated word, I would spell it for them. And then that would be typed and printed up. That's what transcription is. It's the exact message. It's not, you kind of got to do it this way. That's what transcription is. So DNA making RNA, and it makes what's called messenger RNA that carries the message. Those are single strands. They can leave the nucleus and go to ribosomes, which are made of a combination of additional RNA, shaped and made differently, called ribosomal RNA, with proteins. And that ribosome is a platform that receives the messenger RNA, and there's another RNA called transfer RNA that is in the area, and that will latch onto the appropriate amino acids, and all of it is based on a series of codes based on 3 letters. There's only 20 amino acids, 3-letter codes, and you may have seen this. It's called the genetic code. The genetic code is in the language of RNA. In other words, it has the U. So you might have UUU or UAC or GUA, whatever. You have all these different potentials, but they all code for 20 amino acids. So you have a lot of built-in, what we call redundancy. Because of that, understand that's the magic of DNA. We replicate billions of cells every day. We make very few errors. An error in DNA where you don't have the A's and the T's and the G's and the C's matching up has a very familiar name. It's called a mutation. People hear the term mutation, they're like, oh, someone's going to die. Oh my God. No, a mutation is an error in DNA, period. The vast majority of those that occur have no effect at all. We call them silent mutations. There are so many redundancies and safeguards in making our cells that most of those cells that have an error that might be really bad are destroyed anyway. It's almost like it's almost like a suicide pact that happens. You'll learn about it. It's called apoptosis. So they basically destroy themselves before they can start to replicate. Okay. So understand that's a very different process. So all of the steps, messenger RNA going to the ribosome RNA, the transfer RNA, bringing them in the appropriate order, all of those are called translation. We're taking the transcribed message and translating that into an assembly line of polypeptides, or if they're long enough, proteins. It works. And that's basically it. This shows you it. I didn't like playing it because it didn't really, but it just shows you the different bases that are there. And they're showing you how, here's the backbone. You can see the phosphate and the sugars, the P's, and you can see how they're not only attached to one sugar, they're attached to both sugar. That's what I mean, the backbone. And look at the difference. You look at the bottom right, uracil and thymine. They differ by a carbon group. All of those places in science we number. If you're doing organic chemistry, you know the numbers. We don't do the numbers here. Okay. Molecular biology, we do the numbers, but not that. And you can see how similar adenine and guanine are. Adenine has a nitrogenous group in a different position, that's all. And they're showing it to you how they put it together, and you see the numbering there. Thankfully, there's a way, when I had that anti-parallel thing, the strand that's going in the upright direction is assembled in one direction, and the other in the opposite direction. One of them is more efficient than the others. And there are some, not problems, there are little pieces that have to be addressed. And that's something that in more advanced molecular biology we talk about. And so that's really the sum and substance of that. And the very last part of all of this, that thankfully is over, is the most important of all the monomers. Again, the nucleotide is by definition a small molecule. It's called adenosine triphosphate. It involves the A. There's also one called GTP, or guanosine. You won't get to that just yet, but it's coming. That has a lot to do with how signals are carried out from outside the cell to inside the cell. And so, but ATP is by far away the most important molecule that we have. It's remarkable. Every little piece of it we can use for something. But effectively, it's how we store energy that we get from whatever we consume, and we release it by breaking apart these high energy bonds that are made by phosphates being held together scientifically. It looks like that. So here's the adenine. Now we're in the ribose, not the deoxyribose realm, so we got that oxygen over there. Don't have to know where it is. And the phosphates. You can see the phosphates all have a negative charge. So by definition, when you have adenine and ribose, the same thing if this was guanine and ribose, we call this adenosine. Adenosine is a powerful chemical in your body in multiple systems. In general, its major role is as a neurotransmitter. That means it gives instructions to your nervous system. Typically, I know the instruction is going to give my nervous system when I get home. Ooh, you're tired, take it easy. Basically, it'll start to basically create things that will induce you to get closer and closer to sleep, like me talking to you. Didn't have to know it. Just like going, yeah. My wife threatened she's going to come to class and she'll will. So the adenine, the adenosine is like that. Every morning, I don't know about you, I block adenosine receptors. How do you do that? That's the way caffeine works. So whether it's coffee, tea, chocolate, energy drink, Red Bull minus whatever alcoholic intake you might put into it, all of those, you are blocking adenosine receptors. By blocking those receptors, you are stimulating. Wake, alert, whatever, however you want to describe it. There's a variety of other effects. You take too much of it, heart palpitations, take it for too long, and obviously there's probably could wire you pretty badly if you really overindulge. So, I mean, and you do have to be very, very, very watchful about that. That's adenosine. You're going to learn that with one phosphate, it has a lot of activities inside the cell. And the major is this. The reason why the energy is there, these are negatively charged. If you have two kitchen magnets that have the same charge, they'll stick together. If they have opposite charges, they'll stick together. If you have the same, everybody's played with those things. It's almost like you can't try to push them together, and they'll always push their way apart. That's what magnetism does. If you take a third one, it really takes extra energy. That's why it's a high energy bond. We store the energy to hold those bonds open, and we can release a disproportionately larger amount of energy by releasing it, and that's how we energize systems. That phosphate group energizes various things. When we bring sugar, glucose into a cell, when insulin opens the door and sugar comes in, we immediately put a phosphate for energy. That's part of that activation energy, so we can use that sugar eventually to make more ATP. We spend, actually, for every unit of glucose that comes in, we spend about 2 ATPs. And in doing so, when we're all said and done, we generate at least 2 ATPs from the first process alone. Fancy process named, you don't have to know, called glycolysis, splitting the sugar. So everything is done biologically is to try to either conserve energy or put a little in and get a whole lot out. So it's very, good business, so to speak, trying to maximize energy output. And that's all they're just showing you. That means inorganic. And you'll see this again. Why I'm making a big deal? You're going to see as we split that ATP into these two components, when we get to muscle, it's how we reset a muscle so it'll fire again. So all of this has relevance down the road. When we talk about movement across membranes, probably get a little start on that today. What effectively happens, it opens these doorways. But it's basically doing so against the group. We're going to talk about concentration gradient. We have a large amount. This is a class that isn't a very good example. So, I mean, there's supposed to be 13 of us. We got 10 today. 11. That's fine. I'm used to that. But if we had a whole class, eventually all the seats would fill up. You wouldn't have 6 people sitting all in the same row and sitting on top of each other. No. Okay. We call that concentration gradient. Basically, you go from higher concentration to lower concentration. When we have to oppose that and we want to kick something out, we got 6 people here and you want to push them away, and it expends energy, okay, to go away, to go into, let me rephrase it. It's like you're trying to, the crowd's all leaving the ball game and you forgot your sweater and you're trying to get back in. Not easy. Okay, you're expending energy to get through that wall of people, and they're not going to like you when you do that. Nevertheless, you use energy, ATP. When you contract a muscle, you use energy. When you try to put things together and store energy, you have to use energy to create that bond to store energy. Voila. That's the end of this. So the one I sent you is the next PowerPoint. I'll introduce it, get a head start on it, and we'll go from there. No, I did not send you anything when because I wasn't here on Friday. I sent the other classes. I knew we would cover it here. You didn't have to listen to me narrate from being away for our anniversary. I'm entitled. We are. How many years have you been married? That many. And figure out. I haven't been married 50 years, but continuously not even. I think it was 33 and 12, so it's 45 years. Discontinuously. First got married in 78. It's okay. My wife first got married in 79. We were with our same spouses for over 30 years. What were we thinking? I'm terrible. Okay, here we go. Voila. Cells. Probably had this stuff in lab. We'll give it the quickly cell theory. All living things are made out of cells. They're single cell organisms. You'll learn about a micro like bacteria. We're multicellular organisms. So they're all cells, structure and function work together, all good. Continuity of life has a cellular basis, so all cells come from pre-existing cells. Okay. Cell theory 101. Been around, nobody pokes holes in it. Again, in science, when you have a theory, it's very close to being a law. That means people have tried to debunk that, but they can't. You see it more. I think I would present this differently in lab than I do in lecture, because I'll just talk about in lecture the cells that are relevant to what system we are studying. But it gives you an idea of all of these things. So, for instance, in AP2, you'll look at the red blood cell and how it works. You look at the macrophage and how it works. You look at the sperm cell and how it works. You've already looked a little bit at epithelial cells. They're different because they form sheets. They're all tethered together. There's no space between the cells. You studied connective tissue in lab. What connects them? Those fibers. Okay, there's space between the cells, but because they're connected, fibers are connecting. That's where it gets the name, connective tissue. They connect things together, plus the cells themselves. But the cells are generally not tightly packed. Sometimes they are. Again, ligaments and tendons, there's fairly tight packed, but it's mostly fibers in the cells. Muscle cells are so unique. They are a multicellular. They're a confluence of many cells. So we call them multicellular, and they have a very, very specific order. They don't have a lot of other organelles. They only have organelles they need. And those are organelles that are designed to facilitate motion. And we study that. We spend a whole unit studying muscle cells, so I don't need to do any additional presentation, but you'll see it when we get there. And fat cells are storage vehicles. And what happens with fat cells, the fat, basically, the way we would express it, it kind of pushes the nucleus to one side. And over time, when you have it, particularly in certain organs where there's fat cell accumulation, presses on the other cells and damages them, most notoriously the liver. One of the things we see in people with liver disease, let's say hepatitis of some type, or particularly alcoholic hepatitis, which is the most common, alcoholic livers. What happens is those livers, it damages the cells, they're replaced by fatty tissue. Over time, the fat damages surrounding cells, they scar, and the liver begins to shrink and become very rigid. We have a term for that, cirrhosis. And cirrhosis, and a cirrhotic liver, like particularly alcoholic cirrhosis, is a very tragic diagnosis. Because there's not you run out of things to do other than liver transplants. Or shortened lifespans. So fat cells in general, but for you and I, we have fat cells everywhere, around your heart, around your kidneys, around a lot of organs. They're protective in nature. They're supposed to be. Behind your eyeballs, you have a fair amount of fat to cushion them and protect them. White blood cells, very interesting. Very few cells kind of are on their own. White blood cells and red blood cells work in numbers, but they aren't tethered together. Okay? And all those tentacle-like projections, what they are able to latch onto multiple things at a time, like bacteria or viruses. So they have that advantage. And we do a whole unit on nerve cells. And of all of these, the sperm cell and the red blood cell we don't even really necessarily consider them cells. Red blood cell lost its nucleus. So really, as it's matured, it became just elegantly shaped to carry oxygen. Where the sperm only has half the amount, it's what we call haploid. It just has the genetic material from the father. And everything else of that is merely for delivery to the egg, which has everything else. That's the genetic material for the mom and all the cytoplasm. That's why when we talk about DNA testing, okay, a lot of times you're only going to get what we call mitochondrial DNA. That only gives you half the picture. That's like the mom's DNA. So you can see if it's someone who's related, but we can't make a positive identification forensically. So it's a little bit different. So a lot of times forensics is an interesting way to frame some of this stuff. And so here, all that is, a sperm cell is the head up here is the way that it's able to enter the egg. That's the genetic material. And this is the motor. And this is basically the propeller. That's all it is. So it's really designed as just a delivery unit. So there's not a whole lot. So now we get into the cell itself. We'll do the basics that are here. All cells, common structures and functions, plasma membranes, that whole phospholipid bilayer. It was all about plasma membranes, understanding the way proteins folded, how there were areas that were fat soluble and water soluble and charged. All of that is about making membranes. Cholesterol, all of that is about membranes. If we take a phospholipid and put that into water, it will naturally form the very famous bilayer that makes a membrane. And because proteins, if you put proteins in there, some of them would insert themselves into that membrane. And such just the way they do in our membrane. And moreover, if you put cholesterol in there, it would cause that to form really something almost identical to what we have, the very famous fluid mosaic model. Because cholesterol stabilizes it, lets it open and close, is able to hold on to the proteins or allow some proteins to move, or as a platform for reactions. And yet the phospholipid bilayer is ridiculously skinny compared to these things. The proteins are much, much larger. And I mean, I have some good animations that I can show you with this, assuming they're still available online. I think they are. If not, I think you can find a YouTube video. And certainly the nucleus, the main control center, those are the major structures. But there's so many more, and this is a pretty good generic view of not so much any cell, but to give you an idea, I think, of the complexity of the cell. We're going to go over all of those in time, certainly not today. But I mean, well, I was a kid, first time I heard the word cell, I think it was about fourth grade. It takes us before 1960. I don't think any of you were here. Teacher didn't use that marker. Teacher got up there. That's a cell. All we knew, we were sure we were composed of rectangles with little circles and maybe little balls in there of nucleus. That's all we knew. We didn't know all those shapes. It's like everything is so two-dimensional. You have no idea. I have the luxury of going back in that time machine and remembering those things. Which is the stuff that was saying this. That today, with your knowledge, right, just having been educated and being here, we go, aren't you? Below. Again, you thought I was going to say bullshit, but I wouldn't say that in class. But that's what we heard. This gives you a better idea. Karyotic cell like our cell, that's that handout. And indeed, we study it. That's what you looked at under the microscope, or saw the slides projected. Different cells, bone cells don't look the same as blood cells, white cells don't look like red cells, etc., etc. We look at the cellular structure. Indeed, when we analyze and diagnose a disease, okay, we're diagnosing a tumor, whether or not it's cancerous or not. It can't be what looks like a cancer. You have to do it microscopically. I operated for 25 years. When you took something out, you sent it to the lab to be evaluated. That's the way to prove A, what the diagnosis was, and B, that you actually did something. You always tried to make sure you had a certain amount of specimen as part of the process that was there. Okay. So, yeah, the nucleus typically predominates, and we'll go into the structures. But what you can see there, and that's what makes that kind of cell, the eukaryotic cell, is all the membranes. Effectively, when we look at it, it's called the endomembrane system. It's a series of membranes that start, there's the nucleus, you can see the little pores that are there for RNA to get out of, and materials to come in, and materials to wait, building blocks to go in, waste products to flow out. And from that point, anything that comes out of there that's going to be in a membrane or traveling by way of membrane, even spit out from the cell, is encased in membrane to protect it. Inside of the cell is an extraordinarily harsh environment. It's not membrane, it's filled with chemicals, acids, and bases, and chemical reactions going on, salts, and a bunch of stuff that would destabilize, they would denature the protein. They would alter it, certainly. And so effectively what that membrane system, and we look into that and we can determine if you had a liver cell, you have a large amount of something called smooth endoplasmic reticulum. If you had a pancreas cell that was making insulin, you have a large amount of what's called rough endoplasmic reticulum. They have different roles. So that's why, I mean, it's less about if one really looks like that. It gives you an idea that there's more to it than just the shape and the significant degree of complexity in that box circle that we all looked at. We're labeling that. It's like everything was flat. And I was in a school district that considered itself very high-end. Moving on. So we have stuff inside the cell and stuff outside the cell. Okay? Inside the cell and outside the cell. Outside to start, fluids. They're called extracellular fluids. There's a number of subsets. Intracellular, INTRA, means inside. Inter, INTER, when we talk about interstitial fluids, means in between cells. So extracellular is sort of the big term. And then all these other ones are like interstitial between the tissues, is what that means. And so there's always fluid, because if they dry, they don't function. Moisture is inherently, it's why when you get dehydrated, and it's unlike for you and I, for you, getting dehydrated, something you can cope with. When you get the very old and the very young, when they get sick, like vomiting and diarrhea, that's why they're at risk, because they can't tolerate those extremes of dehydration. And that really creates problems. So fluids are paramount there. Plasma is just another fluid. Cerebrospinal fluid, another fluid. They're not identical, they are similar. For instance, where plasma, well, if you spin plasma, typically you only find an occasional white blood cell. They go differently because of their weight. Where cerebrospinal fluid, if you would take a sample of that and find a white blood cell, that's likely that that person has bacterial meningitis. And they are in big trouble. You have to treat that this instant. Not later, or not see what happens overnight. If we don't treat them this instant, they could die within hours. So there's a lot of importance with analysis of these fluids. Joint fluid. We would siphon or take out, a certain amount of joint fluid and send it to the laboratory to determine what kind of arthritis they have. Was it an infection? Was it something else? for instance. And again, saliva and mucus probably differ more by the amount of proteins. Effectively, saliva lubricates and has some chemical properties. Mucus inherently, because of more protein, sticky. What does it do? It traps stuff. Or it provides a layer of mucus because it can guard against harsh chemicals like acids or bases. Acids in the stomach and bases or alkaline in the small intestine. Because the controls in your gastrointestinal tract are all about pH. All about pH. So all of those things, and this is connective tissue. Substance that acts as a glue to hold the cell together. And now the membrane effectively is the barrier between them. And by definition, every one, if we go back and see that cell thing again, every one of those membranes, whether it surrounds the nucleus or a mitochondria or a lysosome or as part of the Golgi apparatus, they all have the same thing associated with them. By definition, they are selectively permeable. Living membranes, that means some stuff can come in and some stuff can't. Some stuff can get in, some stuff can get out, but there's always a way to move stuff in and out. It's not always the same stuff. Semi-permeable, selectively permeable membrane. A phospholipid bilayer with embedded proteins, stabilized by cholesterol molecules, as you will see. You want to call it the cell membrane, you want to call it the plasma membrane, cytoplasmic membrane, all the same. But that membrane is no different than what surrounds a mitochondria or a nucleus. Maybe some structures. Maybe it'll be a double layer like it is with the nucleus, but it's still, they're all flexible. That has to do with the advantage of having the cholesterol. Specialized proteins, they can float because remember, when you fold a protein, there's areas that are compatible with fat. So they can move around. They call it a mosaic because the patterns change that are there. Okay? And you're going to see another one where almost all of these proteins, a fairly substantial amount of them, have sugars protruding. All of our cells, I love this, are sugar coated. That's what's meant by a glycocalyx. Glyco is sugar, calyx is shell. I don't know whether it's plain or peanut, but I'm thinking M&Ms. I'm thinking, hey, and this is a recurrent thing, and that shell has a lot. Sometimes proteins can attach to the shell. Sometimes the shell is for identification, but we all have that. And indeed, down the road, when we characterize things like bacteria, we characterize them by the nature of the shell. If they have a thin shell or a thick shell. Ones that have thick shells sometimes are very, very problematic, particularly in your lung. Tuberculosis would be a perfect example. So in that's why I want to give you context to put the package together. Because, I said every one of these, every AAP1 class today, most of the time, you'd be walking out of here from what I've been doing so far and go, why the heck are we learning? I have to give you context to know going forward where this could be. And some of the stuff, even though I won't test you on it, it doesn't preclude seeing this on an NCLEX. It doesn't preclude seeing this on a licensure exam. Licensure exams are very, very difficult. It's just there's no reinforcement from those things. I think I told you when my wife took hers. She was the first one out. She goes, there's like 20 of us here. I was the first one to leave. And said, your exam is done. For 48 hours, she didn't know if she passed or not. And finally, it was public because you don't know whether you failed or not. They just said, you're done. You either answered enough questions to pass or you bombed enough questions to fail. Fortunately, she was on the pass side. She was an obsessive studier. She was working half time to keep up her benefits and studying endlessly. She's one of those guys. I could test me on that again. Tested you on 6 times today. I want to watch the football game, which we know who won on that battle. Moving on. So it's a bad slide. Here are the phospholipids. Okay. The cholesterol is all through them. They're just showing you, for example, the bilayer. Comparatively, and it's not drawn anywhere near to scale, the sizes are remarkably important. Here, the cholesterol that's embedded, that basically causes, it says further decreases water solubility of the membrane. I'll buy that, but what it does more than anything else, it imparts a certain amount of stability that's there. For instance, one of the things that when someone gets a shot of cortisone for, if grandma's got a bad knee, okay, and she gets a shot of cortisone because her arthritis is acting up, one of the ways that works, because it's a cholesterol derivative, is it stabilizes membranes. And when the membranes, the white blood cells are less likely to spit out chemicals that are nasty or inflammatory. So it has an anti-inflammatory effect. So understand cholesterol has a variety of roles, many of which are useful or cholesterol-like substances. But what it does, it allows you, you'll see down the road, how these membranes can open it up and still maintain a certain degree of intactness, and they're fluid enough to bend in all sorts of different directions. You have proteins that are embedded, and every cell that has a different function has different proteins. Some of them are the same, some of them are vastly different. So for instance, a protein like that has, that's, we call it a channel protein. Nearly every cell in your body has channels for this. That is another name we call it, is an aquaporin, AQUAPORIN. So it's an opening, a pore for water. We have pores in our skin. So imagine the cells, so we always have the ability for water to go in and out. The amount of water that goes in or goes out is governed by the other chemicals that are there. That's what we talk about electro, and I mentioned that before, with vomiting and diarrhea. What happens then, literally the water starts to, there's less water outside the cell, the water from inside the cells tries to balance it out, the cells effectively dehydrate and shrink, and they lose their function. That's why it's so important to rehydrate, particularly. That's why we're constantly watching that. When you treat elderly or very young people, I mean, babies with Pedialyte and all that, whatever they're using today, I don't know. We better be up on it. We're watching the great-grandson Saturday. Nobody here has any great-grandchildren. These are some of those proteins. Different shapes, different roles. Some of them, these are integral proteins. Sometimes they are proteins that protrude outside. Peripheral means they're either on the outside or on the inside. Integral means they're there. Here you can see, and these are relatively simple hexoses. Simple 6 carbons, glucose, fructose, galactose, the ones we learned about before. Those short chains do everything. Things can attach to the chains. They provide identity, as you will see shortly. All of those things are there. So they are the ability for cells to latch on to one another. And so so you can review that as well. And it talks about physical barrier, selective permeability, membrane proteins. What's happening in one cell will influence the others. We'll get to recognition, all of that. It's just the trouble when you have an illustration like that. You could, if you wanted to focus on it, and either you could print it and enlarge it or enlarge it on your screens at home, however you wish to do it. You guys are a little more handy with that stuff than we are. I don't know how to do some of that stuff. I don't bother with this. I don't think there's anything here with it either. Yeah, yeah, and they're basically telling what can get through and what can't. So, and we'll talk about that. Things that are fat compatible can get through the membrane. As long as they're small and they're made out of fats or lipids, they can get through. They can bake, because again, they're not held together per se. They're just next to each other, comfortably providing a barrier. And something like that, whether that was oxygen, easily, that's small, and remember, symmetrical. So oxygen, carbon dioxide, nitrogen, being symmetrical means it's not a polar molecule. So non-polar molecules that are small can get through as well. And that's going to come down as time goes on. Large molecules or charged particles like sodium or glucose cannot. Hydrogen ions cannot. They have to have passageway. I guess water is tiny, but water, there's, again, With rare exceptions, water can move in and out. That's all that they say in that particular animation. So you can look at the animation itself. This gives you an idea, proportion. Okay, so proportionately, 75% phospholipids, and that's the whole idea. They naturally, the hydrophobic hate the water, don't like water, come together in the middle. The hydrophilic ends are on either side. Okay, so the outside of the cell, the extracellular environment, water soluble, charged particles, glucose. The inside of the cell, same deal, just different charged particles, different chemicals, but water soluble. And so that compartmentalization, there are lipids that protrude, lipids with sugar groups on the outer membrane of the surface. Again, sometimes for identification, some of those in different, in some bacteria, those are problematic. And cholesterol, as you can see, stability. And so here it is. This is just the way they form. They drew it in a circular fashion. Nonpolar tails hide, meaning the fat soluble or lipid soluble hide from the water. Okay, whatever. The proteins. 6 of them. Take a break. You with me on that? Yeah. You were ready to take a break 5 minutes ago. An hour and 5 minutes ago. So I sat where you sat. I know I was trying to be I did the transition to teaching from practice. I was going to I couldn't fast track it to go like high school because it pays better. Be like a high school science teacher. And I was 2000, 9/11 happened. So it was 9/11, 2001. I just started. I'm sitting in a classroom at a school where I taught adjunctively, La Roche, over in North Hills. And here's this 50-year-old guy, not quite, 50-year-old guy, basically, and with folks, guys your age. Not so different. Students are students. If you go to community colleges, you have a lot of what are called non-traditional students. Meaning, I enjoyed those classes because they had more diverse experiences and time. So I had people from, particularly trying to become nurses because it was a well-paying field and there's a high demand. 30 to 60 easily. So membrane proteins communicate with the environment. Very large mass. Proteins are heavy. Specialized functions. Some tethered, some flow freely. Integral means they're part of the membrane. Peripheral, sticking out or sticking in. So most of them go all the way across. So they use the fancy term transmembrane. I'm not going to ask you about that kind of stuff. Hydrophobic and hydrophilic, yeah, same thing. Channels and carriers. So this gives you, there's 6 of these things. And you can read this as well as I can read it too, cell-to-cell connections. So if the proteins that stick inside the cell have influence inside the cell, the proteins that stick out outside the cell. And so there's 6 of these that we need to go over. In the interest of having you survive, let's take about 10 minutes and we'll come back. How's that? So it's 4:13, we'll say 4:25. Just give my voice 10 minutes and your attention spans may improve. The old days, we ought to have a cigarette by now, but people don't do that anymore. I think I'll do 2 dictations.